Welcome dear students. In this video we are going to learn about fermentations. As we all know that most eukaryotes are obligate aerobes. That means they grow only in the presence of oxygen. So the term is obligate aerobes. Obligate aerobes. This includes human beings also. Now there is a few eukaryotes are called facultative anaerobes. Facultative anaerobes. These organisms can grow in the presence or absence of oxygen. The example of these organisms like annelids, some mollusk, some yeast. Okay. Now in this class I will describe two types of fermentation, one that is take place in the yeast and one in the mammals. Okay. Now in the both cases the glucose is actually converted into pyruvate by glycolysis. Okay, two pyruvate are produced obviously from glucose. Now in case of yeast, this pyruvate is actually converted into acetaldehyde, and the enzyme that is responsible are called pyruvate decarboxylase. As we all know that decarboxylase enzyme actually removes carbon dioxide, so it will remove a carbon dioxide, and this enzyme actually helps take the help of TPP, thiamine pyrophosphate. Okay, and magnesium also take place in these reactions. Magnesium. Now pyruvate decarboxylase enzyme converts the pyruvate to acetaldehyde. Now the formula of the pyruvate is CH3 C double bond O COOH. This is the pyruvic acid or pyruvate. Now the acetaldehyde, after releasing the COOH, means carbon dioxide, it becomes CH3 C double bond OH. This is the acetaldehyde. Now there is another reactions occur that change this acetaldehyde into ethanol. And here the enzyme is called alcohol dehydrogenase. As we all know that decarboxylase means it removes carbon dioxide and the dehydrogenase means it donates hydrogens. So the hydrogen donor is here the nicotinamide adenine dinucleotide, NADH, nicotinamide adenine dinucleotide hydrate, plus H plus, it becomes NAD plus. So it actually donates the hydrogen, and the alcohol dehydrogenase convert this 2 acetaldehyde into 2 ethanol. The formula is CH3 CH2OH. Now this is the ethanol, and human actually take the advantage of this fermentation that is taking place in the yeast to make beer and wine. Now this is the yeast pathway of the fermentation, is called alcohol fermentation. Now in the mammals, the pyruvate that is produced actually, so there are 2 NADH will be converted into NAD plus. The pyruvate in case of mammals is converted into lactic acid directly, lactic acid. So there should be 2 lactic acid, and the enzyme that actually helps in these conversions are called lactic acid dehydrogenase. Lactic acid dehydrogenase. So there should be also a hydrogen donations, and here also 2 NADH plus H plus actually donate the hydrogen and convert it into NAD plus. Okay, so this is the lactic acid that is produced, so it is called lactic acid fermentations. In muscles that actually contracts vigorously, then during the exercise it could generate much more lactic acid because the scarcity of the oxygens. Okay, now when there is a scarcity in the of oxygens in the muscles, then glucose to pyruvate actually occurs by the glycolysis. Then this enzyme actually gets activated and make lactic acid. Now the lactic acid come out from the muscles to the liver, and this lactic acid is converted into pyruvate another time. And do you know this same enzyme actually convert this lactic acid to pyruvate, and actually NAD plus, now NAD plus actually take the hydrogen to make this pyruvate NADH plus H plus. So this process that is occurs in the liver is called Cori cycle. You remember this, Cori cycle. It's also known as lactic acid cycle. Now when this pyruvate is generated in the liver, this pyruvate can go to the TCA cycle, we all know, tricarboxylic acid cycle, or the pyruvate can be converted into again glucose by gluconeogenesis mechanism or pathway. Now the basic questions here is why during the absence or scarcity of oxygens cells go through these fermentations to produce lactic acid. The answer is in this electron transport chain, because in the electron transport chain there is a complex called complex 4, it's also called cytochrome C oxidase. This complex 4 is actually transfer the electron to the ultimate electron acceptor. You have to understand this. I am not going to details because I will take a separate class on electron transport chain. In the electron transport chain there is a complex called complex 4, that is also known as cytochrome C oxidase. This is the last complex where the electron actually transported, and the ultimate electron acceptor is the molecular oxygens. You have to understand this. As there is no oxygen, scarcity of oxygens, then the electrons that is actually passed through this NADH because they carry electron. So this electron cannot be delivered to the oxygen because there is a scarcity of this oxygen. So this NADH actually remove its electron and hydrogen to this acetaldehyde to ethanol to make ethanol. And also in case of lactic acid productions, this NADH actually donate this electron because it cannot donate the ultimate electron acceptor oxygens.